Now it's my esteemed pleasure to welcome Mark Hamer. Mark is a laboratory scientist and technician at the BRC, and he's been with us for a bit over a year now. And he's thoroughly familiar with the CyTOF. He's our top manager of the CyTOF and the experimental applications of mass cytometry. And he's on basically the manager of our expanding facility. And he's going to give you a quick overview of how the machine operates and show you some data and things that we can extract from samples just from our initial work with the CyTOF. Over to you, Mark.
Okay, so in framing the context of mass cytometry, I think that it's important to look at how today's technologies are approaching scientific problems, specifically when characterizing and quantifying protein expression on live cells. We've reached a stage now where we're really able to dig into cell-cell interactions, really unique, rare populations within blood, etc. And so when we start approaching these kind of questions, it's good to, I think, redefine resolution and what that means in the context of resolving cell attributes or phenotypes. So I've kind of put 4 here. I think that these cover most of the bases. Maybe somebody will have another one that they can bring up down the line. But what I thought of here is basically throughput, the number of cells that a technology is capable of analyzing in a given time, the number of parameters measured, or the dimensionality, which is a key point that we'll come back to again and again in this presentation, and the capacity or sensitivity to detect the metric being measured. So with fluorescence cytometry, this would be the sensitivity of the detector to the fluorophore. In mass cytometry, this is the sensitivity of the detector to the actual mass of the atom. And then there are the traditional, we can't forget about pixelation when we're looking at the optics that go into the more traditional framework of resolution, especially with microscopy. So that said, I want to give a brief overview of what my talk today will cover. I'll start with a kind of comparison between flow cytometry and mass cytometry. I'll then lead that into an introduction into our CyTOF 2 mass cytometer, and then give a general overview of the workflow and data analysis that comes with running a CyTOF experiment. So everybody, I think, in this context should be fairly familiar with immunofluorescence in terms of flow cytometry, but I'll quickly kind of go over the strengths and limitations that are relevant to the technology, especially in regards to deciding whether or not mass cytometry is something that should be considered. So the strengths of immunofluorescence cytometry, first of all, this is the gold standard. So everybody uses flow cytometry, myself included. It's a very powerful tool with a huge amount of support in the field. Also, the acquisition rate, especially for just analyzing cells, is very high. It's over 10 times higher than you'll see those of mass cytometry. It's quite cost effective for small samples, again, due to the availability and kind of history of commercialization in this sector. And then because of that, also, we have a very robust system for support. And last but not least, I guess this is the really awesome thing that I continue to be jealous about flow cytometry, especially when I'm atomizing my cells, is that the cells can be recovered in flow cytometry. So sorting is still a very defining and useful tool for flow. That said, we do have limitations with the technology, specifically when we start to increase the dimensionality of the parameters or markers that we're interested in. So I don't know, myself, I've never done more than an 8-color panel. I know that there are people constantly out there claiming that they can do 15, 20, but we know that these become extremely difficult to compensate, and specifically because of this spectral overlap that occurs at when you start adding colors and fluors into these experiments. And then there's also issues of autofluorescence, which limit the capacity to use flow cytometry for things like intracellular phosphostaining. So I have a very brief slide here we'll see how the animation works out, to introduce just the idea of spectral overlap here. And this is just a simple 2-color system looking at FITC and PE. And I just want to use this to kind of demonstrate how we have to create bandpass filters on immunofluorescent cytometry detectors in order to actually limit the spillover from the tail ends of these other floors. So you can see here we have, you know, two very nice bandpass filters set, but you can observe that if you were to increase this to 12 colors, 10 colors, these start to get very crowded, and you actually start to deteriorate the quality of your signal as you go. So an approach, actually a Canadian approach originally in Toronto to this problem was to, rather than conjugate antibodies to fluorophores, conjugate these your antibodies to lanthanide group metals. These are the rare earth metals. And what this allows for is building really complex panels of up to 50 parameters with very little concern for spillover, compensation, autofluorescence, things like this. This also leads us to kind of something that I struggle with kind of getting the point across, is that the cost of designing a large panel for CyTOF ends up actually making a lot of sense when you compare it to a similar panel designed in Flow. Our conjugations are done in-house, which keep the costs down, and this enables us to really build nice panels that are cost-effective, especially when you start reaching over 30 parameters. Additionally, we have the capacity to barcode samples, which I don't want to focus on too much. It's a technique that we've struggled with a little bit in our studies so far, but it does allow you to essentially use a series of metals to create a 3 of 6 barcoding for cell samples. And what this does is you basically stain the cells, each sample up to 20 samples, with a series of these 3 isotopes, again 3 out of 6 total, and then you can deconvolve that data downstream during the analysis to actually pick out your panel, or pick out your sample, individual samples. And the power in this is that you are able to stain all 20 of your samples after barcoding at the same time, and so you really minimize the amount of heterogeneity in your staining that can be a big issue, especially with large panels like this, where it's quite easy to introduce small technical variations. And also, like I mentioned, the lack of autofluorescence means that intracellular phosphostaining really shines through using mass cytometry. So it's another kind of focal point for this technology. Of course, this isn't all fine and dandy. We do have some limitations that come with the technology, specifically the slower acquisition rate. So we're limited right now to about 500 cells per second. I would argue this is still pretty good when you're analyzing 50 parameters at a time, but it is, you know, about 10-fold slower than your typical sorting or flow experiment. We also do lose cells during the acquisition. As I'll point out here in a moment, the cells are actually atomized in order to gain the information downstream using the mass spec. So there's no coming back from this in the end. Also, the upfront cost can, and this is, like I said, be something that may put people off initially, but the important thing to realize is that you build a panel and you have that panel for hundreds of experiments from then on, or hundreds of samples. And so the upfront cost can seem extreme, but the actual running costs after that is very competitive with Flow. And I would argue that even the panel building is competitive when you consider how many parameters you're building into these. You guys don't need to worry too much about single instrument upkeep. That's my job. It's quite a bit of work to keep these things running. It's, as you can imagine, it's a fluidic system hooked up to a mass spec, which is always a lot of fun to take apart and keep operational. And lastly, I do want to comment about the kind of historical control that Fluidigm, who has been very gracious in their assistance with keeping this machine up and running, but also limiting in the capacity that they have a lot of control over the technology and reagents themselves. So this is starting to change. We have actually additional Flow mass cytometers that are coming to the market from Beckman Coulter, I believe, and other companies. But so I think we're starting to see a shift there. Let's see. So to kind of round this out, I want to bring us back to that idea of resolution. So fluorescence-based cytometry basically favors a type of throughput resolution over dimensionality. Sensitivity can be thought of as the same, basically across mass cytometry and fluorescence cytometry. Anybody really familiar knows that with the brightest fluors, you can actually get a little more sensitivity out of flow, but that's kind of a non-issue in most cases. So really what mass cytometry gives us is a medium throughput, medium dimensionality, medium sensitivity resolution, which I think is unique in terms of profiling single cells. So this makes it an excellent approach for monitoring systems that are complex and contain hundreds of cell types like that of the immune system and complex solid tissues such as tumors and other complicated solid tissues. So how does a CyTOF work? Well, we have a CyTOF here. This is what's sitting upstairs in the Biomedical Research Center. And basically, I want to just give a brief kind of overview of how samples are run through this. I'll talk about staining in the next slide. It's kind of tricky, you know, what point you talk about staining versus running these samples. But basically, what you start with is a cell suspension that has been stained with these antibodies conjugated to lanthanide metals. These cell suspensions are fed through a nebulizer, which basically aerosolizes these cells into single droplets containing single cells. That's critical. This is all driven by a fluidic system built into the instrument. These single cell droplets are then fed into a plasma torch that is charged by a RF generator, which essentially atomizes and then ionizes each one of these single cell clouds. And what comes out is this big cloud of ions and atoms containing not just the reporter atoms that you've stained with, but also all of the common elements that you would expect, your carbon and nitrogen, phosphorus, etc. To that end, a quadrupole is used to actually remove all those lighter weight ions. This essentially puts a slight spin on these charged particles, and the charged particles kind of spin out of control and come out, while the heavier ions are not as affected by the magnetism and are passed through the quadrupole. They then end up in the mass spectrometer, the time-of-flight mass spec, where the heavier ions are deflected at a slower rate, or at or deflected in a longer path than the lighter ions, and then detected here and digitized into a signal for each cell event. So what you see here is basically cells that each have characteristic mass profiles. And these are converted then in turn to an FCS file, which is really the critical step, because a lot of the tools that we use are the same tools that would be used for flow data. And that's, I think, something that makes getting into this kind of data analysis really helpful, is that it's not a completely new way of looking at data. We're very familiar with the format. It's just the fact that your little X and Y axes now have 40 parameters instead of 10. So I said that I'd briefly talk about staining. Really, there's not too much to talk about here. The overall protocol for this is very similar to flow cytometry. A few more washes because of the need to remove any sort of metals that may exist from the sample tubes or anything like that. There's also, if necessary, we will do a fixed perm step for intracellular staining. This is so that we can actually get the antibodies into the cells and mark those phosphoproteins. Additionally, we have a series of pretty smart DNA intercalators that we use to identify both singlets versus doublets and discriminate intact and live cells from dead cells. Those are some really nice kind of kinetics-based intercalators that lend themselves really well to this problem of not really having forward scatter, side scatter like we do on flow cytometry. And lastly, just before the run, we do spike in a solution of equilibration beads, and this helps us to normalize the mass signal across samples and experiments as we continue. Okay, so briefly, the typical mass cytometry workflow. I really like this chart down here. My job is only about 10% of the work. that needs to go into this. That's not really true because I'm also involved in these other aspects. But the critical thing to realize here is that 90% of the work comes in the experimental design, the reagent validation, and the data analysis. The operating of the machine and collecting of data itself is very straightforward. So I'm going to actually talk about these segments here, in particularly the experimental design, the panel validation, and then the collection of data and analysis of it downstream. So something that I've recently started to really emphasize when planning experiments with people, especially with clinical samples, is the need to have consistent sample processing. The preparation of these samples is very key. So you can see here that this is a sample that we recently received. These are cord blood samples, and you can see that when you're looking for small differences in heterogeneous human populations, you hope that even the color of your samples would be the same when you go into staining and things like that. Obviously, color is by no means a metric for sample quality, but I think it just goes to show that these samples were clearly processed differently in some way. Next, the decision to either use a validated off-the-shelf panel, of which we have a few here at UBC, versus a custom panel is the next decision that needs to be made. So we, like I said, do have kind of base panels that we use, and we build upon these, kind of inserting and removing different markers depending on the question being asked. So we're certainly able to modify these more easily than building from the ground up, but there is still a possibility if you have a really unique system that you're looking at. Some of these common markers that you would expect to find in a panel are those of like T cells and B cells, monocytes, macrophages, etc. So we have probably, I would say, a core of about 20 antibodies that we keep on hand and in stock that we really go through quickly as our indexing type proteins. The next point I'd like to make is that when actually designing a custom conjugation or a custom panel, it's important to realize that the mass spec itself does have a peak sensitivity range that basically falls between this 150 and 170 AMU range. And so what's important to take into consideration when designing your panel is that you really don't need to have your CD8s and your CD4s out here in peak sensitivity. You want to save that for markers that are maybe low abundance or that are more important to the experimental outcome. And that's obviously something that would be discussed on a case-by-case basis during the panel design. Next, although I said earlier that there is no spillover, and that is for the most part true, we do have some considerations that we need to make, especially in terms of oxidation and isotopic impurity. Some of these metals, although 99% pure, they will contain a 1% impurity in a metal or in an isotope just +1, for example, AMU more. And so this is really kind of my job to pay attention to these kind of things. But I guess when it comes to panel design, really what you want to make sure is that, again, you don't put a really high abundance marker in one of these metals that is known to be isotopically impure, because that could potentially spill over into a mass channel below or one above. To give you an example of a panel validation experiment, this is one that I actually recently completed for Kelly. And basically what we did, we had a few markers that we wanted to add to an existing panel. And in order to validate that, what I do to ensure that these markers or that the antibodies are working properly is I'll take a cocktail of cells, and I'm certainly willing to grow up new or receive new cell lines to test this. But basically grow up and then create a cocktail of cells that you know will express the markers that you're inserting and validating at the time of testing. And so what we have here is a series of 5 cell, or a pool of 5 cell types that we then tested. And we were looking, mostly this is actually validating a CD34 antibody that wasn't working, as well as a IL-17 receptor antibody. And what we see is that this is kind of some downstream post-analysis work using clustering algorithm. So I'll get to that in a moment. But what we saw was that we were really able to discern unique cell populations by certain markers that we were testing. So we really like to validate that. And of course, before this, you would look at the biaxial plots also to ensure that those actually show a shift in signal or a real signal. So to get to that point of actually having those nice colored t-SNE plots, you actually have to do a bit of cleaning of the data prior to analysis. This involves firstly a debarcoding step if barcoded. I'm going to leave that for now. But more importantly, a normalization, which if you notice, there's a fairly linear degradation of signal intensity as you follow an experiment temporally. And what we tend to do is reach a certain point at which we will retune the machine. And this brings the sensitivity back up. But if you don't control for that, you essentially see a spike in your signal and all of your samples run after that tune, so especially in comparison to the samples just prior. And so we have a nice MATLAB script that, and actually there's also a nice R script now that runs this normalization really quickly, and it just kind of levels out the signal across all of your markers based on these equilibration beads that we spiked into the samples initially. We then go through some simple cleanup. This is much like you would perform in flow cytometry. So we have single nucleated cells based on these intercalating agents. We then gate out these dead cells using cisplatin, which binds preferentially to dead cells, or kinetically preferentially to dead cells, leaving you with a population of cells, of live cells, that is lower signal than those of the than those positive. And then we remove the beads from our population. You obviously don't want to have a bunch of beads when you go into data analysis. That gets us to the really fun thing, and I think that this is something that Gary Nolan at Stanford introduced as a really interesting problem, and I really love his the way that he presented this issue. When you look at a regular two-parameter biaxial plot, it's really simple to make a inference about what you're looking at. The same is true even if you add 3 parameters. You get 3 plots. It's really easy to compare these even across samples. However, when you increase to what I would say are the standard limits of flow cytometry, and I'm sure some people are shaking their heads here, but up to 9 parameters, you increase this to 36 plots. And this, while okay, yeah, sure, you can go through and look at 36 plots. Now you compare this across 10 samples and you're looking at over 300 plots. And comparison amongst those gets tedious, if nothing else. Now, in mass cytometry, we actually have over 30 parameters in a typical experiment, which means that you have 500 plots per sample that you would then need to compare across samples in a given experiment. So this really becomes, I would say, an impossible task. It's certainly valid to look at markers within those of interest and look at the biaxials to kind of inform decisions or to inform something that you find in the later data analysis, but going through and simply looking at these plots one by one is impossible. And so what we've implemented here is a pipeline of various tools, including MATLAB, R, and Saito Bank, each having kind of unique features that we've found useful for different applications. R is obviously kind of our workhorse. We do a lot of our analysis here, mostly because it's free, and the amount of support for new technologies is really kind of leaps and bounds above something like a paid software, for example, Saito Bank or MATLAB, where there's a lot of very powerful data analysis tools that can be used, but learning the syntax is a little trickier, especially for biologists who are more familiar with R. So I want to briefly kind of run through one of these packages. This is CyTOF Kit. It's a phenograph-based dimensional reduction software that runs within the R framework. And essentially what happens here is you feed in your FCS files. This is all done in a graphical user interface, which is really handy for people, again, that can load R onto their computers but may not be really comfortable with working with the syntax or actual scripting. And so you can load these files in, you transform and merge appropriate files, creating a cell and marker matrix. You then run through a cell subset detection algorithm. Specifically, we use FlowSOM and Phenograph. FlowSOM is generated particularly for CyTOF data, and I believe Phenograph was also. So these are both really, really good softwares for actually looking at the interrelatedness between cells and then doing this kind of clustering downstream. These clusters are visualized in t-SNE plots for the most part, which just gives us a nice way of plotting out these the relationship data of the clustering algorithms run prior. We then have a series of relatedness or hierarchical analysis tools, which I will be honest, I don't use a lot of those in CyTOF kit. They tend to be very computationally intense. And so what we actually use for those is another program, which I'll briefly run over here in a moment. But I do also want to note, I just found this out actually as I was preparing this presentation. CyTOF kit is now available as a standalone web app. So this runs in Java on your web browser, and this makes it even more handy to use. Really kind of streamlines the process. So I mentioned another way of kind of building hierarchical clusters and trees. One example of this is Saito Bank. This is a technology or a program that is initially designed in MATLAB, but now purchased and used by Saito Bank, which is a web-based platform. And essentially what this allows you to do is build these spanning network trees. And the color here represents the expression of a given marker, and the size of these nodes represent, or the nodes themselves represent cell populations, and the size of the node represents the number of cells within that population. So really useful, and you can compare the spade trees from one sample to another. So it gives you a really nice way of kind of catching differences that exist in populations that this pulls out. I want to kind of acknowledge some of the additional ongoing projects that we have before we continue. This should give an idea of kind of the capacity that we're capable of here with using the CyTOF. We have multiple blood samples. PBMCs really are the easiest suspension type tissue to approach with CyTOF, but that doesn't limit us from looking at cancer cell lines, solid tissues in terms of kidney, skeletal muscle, spleen, lung, heart. These are all projects that we have in the works currently. And this is true for both mouse and human. So I want to give thanks here to Kelly McNagny, whose name I misspelled. Oops. Kelly McNagny sitting right next to me, and Fabio Rossi, who's been really helpful in getting me going with the CyTOF. Melina Messing, Kelly's graduate student, who also has been very helpful in her work, especially the experimental design. Josh Skrull, he's a PhD candidate working with bioinformatics, that's been really helpful with getting some of the MATLAB scripts running. And then the Rossi McNagny lab, Andy Johnson of the BC Flow, and Mike Williams, our in-house antibody conjugation master. So with that, I want to say thank you to the rest of you and open up the question and answer session.
The first question comes from Stephen Booth, who asks,
Hi, Steve.
Who says, "Hi, Mark. Is it possible to stain intracellular cytokines and phosphoproteins in the same sample? Do these require different prep?" And there's another message that says, "Steve from St. Paul's, by the way, cheers."
Yeah, hi, Steve, and thank you for your question. It is certainly possible to do intracellular and phosphostain in the same samples as you would do your normal surface staining. The only caveat to that is that there can be effects of the actual fixation process on the surface chemistry of your surface proteins, and so you may find that I think we generally run into maybe one marker that doesn't work after fixation, and that can be a headache. But it's something that if you're really interested in doing phosphostaining, can make it worth it to kind of find an alternate marker for that sample.
So just as a follow-up to that, Mark, do you know if you have to use buffers that have phosphatase inhibitors and things like that when you're trying to pick up reagents or pick up phosphomodifications on proteins? Or?
So because they're fixed, it's not really critical to do that. And really, the fixation, you can use a very light fixative, and we actually do methanol permeabilization. So this keeps things relatively not harsh in terms of the preparation. So I'd say that.
Mark, how did you find barcoding for samples? Do you find that it changes sensitivity? Do you find it useful? Is it a problem for large numbers of cells or small numbers of cells? I mean, to me, it sounds like a terrific technique for having side-by-side comparison, everything stained in the same tube to make sure you get really uniform readout. But I'm sure there's a cost at that, right?
Yeah, it certainly is very useful and this is a big if you've designed your panel from the beginning with using barcoding in mind. Another kind of thing to keep in mind here is that barcoding, the way that these kits are designed and shipped from Fluidigm, is you need to have a minimum of 1 million cells per sample. And this can limit really precious samples from being analyzed using barcoding. So there are some limitations there. However, if you design your panel from the ground up with barcoding and when I say barcoding, I basically mean with fixing in mind, and you get your panel validated properly with that fixation in place, it's an amazing tool for not just for staining samples altogether, but essentially what it also allows is an additional doublet removal tool, because you essentially know that each cell that has been stained with a unique 3 metal barcode cannot express an additional metal on top of that. So you really cut out the risk of doublets, which can still be an issue with messy samples.
How about minimum number of cells per sample without barcoding?
How careful are you? So it really depends on your ability to come through the staining process with enough cells. So there are quite a few, I think 6 or 7 centrifugations at a minimum in terms of washing and rinsing after the staining. So it's really kind of making sure that you have enough cells. If you have a population of cells that you have a ton of, you don't worry about those lost cells. But if you're starting with a small population, you can start to run into issues towards the end of losing viable cells.
And then, but Mark, there should be techniques for helping with that, right? You can add carrier cells that you've pre-labeled as
Sure.
a way of trying to, make sure that you keep enough of your cells if cell number is limiting and you don't lose them in the process.
And this is an excellent opportunity to actually use barcoding too. If you've designed this into your panel, you barcode your samples of interest at the very beginning, and then you spike those in with a ton of carrier cells, just peripheral blood mononuclear cells, for example, and you can increase, then you have 10 million cells in your sample, and you can increase the length of your run, obviously, but then go back and remove those cells post hoc and analyze only those that were barcoded initially.
So another one for me is I know that some of the imaging tools, like the TISNI plots, look a lot like what we use for single-cell RNA-seq profiles, which is actually really helpful because it makes it user-friendly for people who are more used to looking at RNA sequencing data rather than flow data. So it seems like mass spec is being used a bit like a medium throughput analysis. With single-cell sequencing, you get thousands of transcripts, and you have to analyze that with your visual cortex, so the TISNI plots help with that. But it seems like the same tool can be used by mass spec.
Yeah, so a lot of these single-cell RNA-seq analysis platforms rely on dimensional reduction as well, and that's what you see. Usually they are relying more on PCE, principal component analysis, which is a linear method of dimensional reduction, and so it's not quite as useful for some of this nonlinear protein quantification, but it certainly outputs in a very similar fashion. You kind of have these clusters that are represented in this either TISNI or, in the case of RNA-seq data, PCA space. Those axes don't mean anything other than for giving you a frame of reference that basically gives you a 2-dimensional best fit view of the highly dimensional data.
Very good. Another one that I always ran into trouble with is for intracellular staining. It used to be my nightmare for flow because you get so much kind of background fluorescence from actin cytoskeleton and other intracellular proteins. And I always likened it to, thinking about the number of proteins on the surface of a sphere to the number of proteins inside a sphere. You really go up exponentially, which gives you a lot of background. So how is CyTOF able to deal with that kind of problem?
Well, I think the really, simple answer to that is you don't have autofluorescence. You're not relying on an optical measurement. You're relying on a rare earth metal that does not exist in a biological system. And so because that is now your metric or your method of quantifying proteins within the cell, you don't worry about actin cytoskeleton, for example, autofluorescing in your FITC channel. It's just a non-issue.
Cool. And then just by way of another comparison, so looking at RNA expression, I'm going to guess, again, based on history, that protein expression and RNA expression probably don't always match all that well. And is there any kind of, any lore out there about if you're doing single cell sequencing on a population of mast cells or stem cells or whatever you're interested in, and you try to compare that with the protein expression that you see by antibody, is there a rule of thumb for how well those things tend to correlate?
I think Kelly's asking a leading question here. Something that I'm involved in right now is building up a new technology here at UBC called CytSeq. And this is a technique that essentially uses oligonucleotide tags to stain proteins. So basically, again, we're substituting these metal or fluorophore tags with oligonucleotide sequences, and we then stain cells with that. And what's really awesome about CytSeq is the ability to stain samples. You can use hundreds of antibodies at a time because you're limited by basically 10 to the 10th number of oligonucleotide as in terms of identifying your protein downstream. And then you still get to run through your sample with your normal single-cell RNA-seq prep. So you get both the proteomic, and I'm calling this proteomic now, surface proteome. You get both proteomic and transcriptomic data at the same time. So this is something that we've recently been funded for by the Faculty of Medicine here at UBC, and we are working on. So
very good.
That will help answer those questions, I think.
And to the people who are afraid of doing CyTOF, you mentioned that actually it turns out to be cost effective. And is that because you're getting from one stain 50 antibodies as opposed to having to do 50 single stains by normal
or building these kind of overlapping panels, and there are a ton of issues that come up with running multiple overlapping panels, and they can be overcome certainly, but it's a lot easier to just throw them all on one sample, do the run all on one machine, one instrument at one time, and gain all of your data at the same time. So I think that the cost, CyTOF is certainly a little more expensive right now. I don't want to allude to the fact that it's not expensive, but I think that what shocks people sometimes is the upfront cost of building a panel, which may not even be necessary if you use a panel that's already been designed.
Fantastic. And then the last question for me is, I've seen a lot of debate on whether it's most efficient to use CyTOF as a discovery tool or as an analytical tool. So for example, if you had, 100 patients that you wanted to characterize, would it be better to use CyTOF to try to identify, with 50 antibodies in that CyTOF discovery experiment, the 10 that are going to be useful for distinguishing differences between patient and normal? Or do you think it would be more efficient to use CyTOF for analyzing a whole 100 patients and get all of that information? What do you think?
I think that CyTOF can certainly be used as a discovery tool, but it's so critical that you have a properly validated panel before moving into that. So making sure that your markers, especially the ones that you think, oh, we're going to find this novel rare cell, stem cell population in the blood that nobody's ever seen before, and we have this marker that we know is going to be on there, you better be sure that that marker actually works and show me a flow chart or something where you see it working. So, but absolutely, it can be used as a discovery tool if you have that information.
That's it for me.
All right, well, Chris Carlson is chipping in again with a couple of questions, so I'll read those out. Let's say your main questions involve tradition cells, for example, just all PMNs, but also interested in rare cells. How does CyTOF compare to traditional flow for the traditional bread and butter cells?
So if I'm understanding that correctly, it's just a kind of broad characterization of common markers.
I guess what Chris is asking is if you know you're interested in PMNs, like neutrophils, and you want to, which do you think is better, CyTOF?
If you're just trying to quantify, get a quick result with, and you don't need the dimensionality, go with flow.
Yeah.
It would be a waste of time overall because the acquisition rate itself is just so slow with mass cytometry. Now, if you have a unique population within those PMNs that you think you're really trying to go after, and you have these additional 8 markers that you want to throw in your panel, then CyTOF starts to become a more viable option.
Well, and I guess if you know that a patient's disease is due to a change in the granulocytes in the PMNs, then just as a discovery tool, if you can throw 50 additional antibodies, and you could pick, ones that are always expressed by PMNs, I bet you'll be able to pull out differences in those patients, especially if you know that they're the cause of the disease.
Sure. Yeah, I would agree with that. And like I said, the key is really making sure that those that the antibodies you're using are validated.
Okay, good. Hi guys, a little bit loud here, but I'll try to be clear. Thanks again, Mark and Kelly. Yeah, I think you guys pretty much got it, and Kelly understands the situation in my lab, which is why he kind of paraphrased. Thank you, Kelly. But just to be clear, yeah, I think you answered it. It's just like we don't want to sacrifice anything on the main cells. Like we want to be 100% sure. Say, for example, we count all eosinophils and basic markers of eosinophil activation, and that's our top priority. But at the same time, saying like, oh, this is really neat to get these novel subtypes of cells, etc. And so the way I put that together is I don't want to sacrifice anything on the traditional things that we need to get, like say an activated eosinophil, but I also want to get that additional information. And if I do this through CyTOF, what are the risks that I'm going to be losing some fidelity or some, that I'm definitively looking at an activated eosinophil where I'm pretty confident with my traditional flow methods, I'm not going to lose that.
Yeah, I think if you're using those same antibodies and you just conjugate them to metals and you design the panel around that being your top priority, you can certainly then use the available channels outside to do the more discovery-based stuff while retaining really strong, consistent data with what you see in your flow.
Yeah.
Okay,
I would agree with that, Chris. And I think I would look at it 2 ways. One is if you're buying an off-the-shelf panel from Fluidigm or from one of the other companies, you're going to get a validated panel that's going to work, tell you everything that you want to know for the standard markers, with much higher dimensionality. Because don't forget, 50 by 50. Think of all the 2-color plots you'd have to look at to get that information with a standard flow panel. So you'll get all that extra information, and especially if you've got a patient population that is different, that'll be huge. But perhaps the more interesting one would be, rather than buying the off-the-shelf, if you buy the off-the-shelf and then you spike it with 5 or 6 antibodies that you're particularly interested in, then you get data that nobody else in the world has, right? You've combined 50 antibodies plus your own favorite 3 or 4, right? That's when you start discovering new populations of cells that you didn't know existed, and I think that's really the power of the CyTOF.
And I'll just add to that really quickly, because this is an excellent way to make this point, is that you'll see in your, after doing the analysis and the dimensional reduction, looking at your phenograph plots and stuff like that, you'll see those populations of cells that you expect. They will cluster together and you'll see those consistently from sample to sample. And what you will notice is that if you really do find this a unique population, that will separate out and cluster on its own, given that it's a strong enough read, and it really provides some nice visualization of the changes that you see in your system.
I guess another thing that people are considering, and it's something that I'm more and more interested in, is using CyTOF for analyzing things other than blood. So for thinking for allergen, if you could have enough, a panel of 24 or 50 antibodies to look at epithelial cells or mesenchymal cells that are coming from brushings, that would be phenomenal. And I think that's really something that traditionally has been done by standard histology and trying to throw, 4 or 5 antibodies in the histological section, which, that's incredibly limiting. It's slow. It doesn't give you new populations quickly. And I think that's a big black box that hasn't been explored very much yet.
Hey, I'll try one more time. I was telling Marshall that maybe it's too loud, but on that last one, and I'm sorry, I didn't see any other questions, so even though I'm asking a lot of questions, I guess it's okay. On the epithelial cell issue, or whether it's that or BAL, et cetera, things we've kind of talked a little bit about, Kelly, the issue, as I'm sure you know, is if you need 1 million cells and you've got other endpoints, it's really tough. But, in the last time I talked, even without the barcoding, and Mark didn't quite say exactly today, but I still remember the ballpark being like 1 million cells. But if you can get it down to like 500,000, then it's a lot more viable for, at least kind of in my hands or my lab's hands, because on a brush we might get just about 1 million cells if we're lucky, depending on the exact brush. And if I can at least have half of those to do some other things, then it's a lot more attractive to give 500,000 to CyTOF.
So my gut feeling, and I'll let Mark answer afterwards, but my gut feeling is we can do that now, Chris. And as Mark pointed out, if you spike with, a cell line as carrier to make sure you don't lose the cells during the wash, I would think 500,000 cells would be easy to do. And remember that with that 500,000 cells, you're basically getting 24 antibody stains or 50 antibody stains, depending on how big your panel is. And in order to do that by standard flow, you'd never be able to do that with 500,000 cells, right?
I hear you. Thanks.
Yeah, and I agree 100%. I think the 500,000 cells, you certainly, the issue that you run into is the barcoding really doesn't work well outside of its suggested range. And we've tested this, we've tried to get it to work, but it just, for whatever reason, it has a range in it, and that's the range at which it works.
Without barcoding?
Without barcoding, certainly you could, 500,000 cells is a lot of cells still, and I imagine that you could still get some data out of that.
Thank you very much, guys.
Thank you.
Thanks, Chris. Nope, I'd just like to thank everybody for tuning in. I think this has been very informative, and please let us know how you feel about the webinar. I think from our point of view, this is a great way of communicating with everyone in the network. And if you have questions that you'd like to submit later, or you have comments on how we've run this and would like to see some changes, please do suggest that we're open to suggestions.